more concentrated than the dideoxys on purpose. So, and I don't know if it's like 10 times or 100 times more concentrated, but yeah, you can fiddle with that ratio if you're having trouble with early truncations. You can fiddle with that ratio if it's reading way too long too, so that you're missing steps. Yeah, since the radionucleotides are in excess, it's not likely that you'll always terminate two bases in every time. Okay, so that cannot be extended. So then what you do is you separate out all the copies which are truncated at every single possible length. And then you read the sequence.
What does truncated mean?
Broken off or stopped. Think of this as terminated. Or shorter than expected. The reading can be by gel electrophoresis. And if that's the case, this requires 4 separate reactions, each with a different dideoxynucleotide, like I drew above for the 4 different test tubes. This can also be done by capillary electrophoresis. With only one PCR reaction where each dideoxynucleotide has a different fluorescent marker on it.
So the first one we got like 16 then?
Excuse me?
You have 1 and 4, so it's 16.
Well, for the gel electrophoresis, you're going to have 4 lanes on the gel that you're looking at.
Oh.
One for the A's, one for the C's, one for the G's, and one for the T's. Capillary electrophoresis, this is a column, it's just one column.
And then you flip on different lights.
Yeah. So, yeah, picture time. Okay.
What do you mean? There's a possible way.
Your dideoxys get randomly incorporated, and so ideally, if your missing reaction works well, then every single possible position for an A will get some percent truncated at that position where the dideoxy is incorporated. So you don't, when you do the PCR reaction, if it stops at the first A every time and you can never extend it, then you're never going to get a sequence. And so you have to have regular MTPs that get incorporated in most of the positions, but then every once in a while a dideoxy gets put in and that particular fragment of DNA is then stopped, it's terminated. And so the idea is to get every single possible length of DNA that stops for a particular nucleotide. Okay, can we look at the pictures?
Yeah.
Yes. So, all good questions, but the picture is going to make this easier to, oops, sorry, I wrote down there. Okay. So this is just showing the extension. That's my template DNA, and this is what I'm extending with a primer, yes.
I have a question about the primer. If you're doing a sequence of DNA that you've never done before, how do you know what primer to use?
You have to have something that is unique to it. So you have to know something, and there are a lot of promoters out there that they look the same from one place to another, or it could be like a ribosome binding site that is a conserved sequence that you know all your genes are going to have that in front of them. So there are little primers out there that you can use that similar for all genes. Okay, so this is our template. I have a little primer for my PCR reaction, and then I'm incorporating my NTPs. And so these are just my normal NTPs that have a 3' hydroxyl on them, so I can link them up as I go. And this is done 5' to 3'. And then every once in a while, I get one that comes in where I'm missing the hydroxyl group. And if I'm missing that hydroxyl group, then I can't extend it. So when you set up your 4 different reactions, you have all the regular NTPs and then one spiked dideoxy. So this would be one PCR tube, and this would be another PCR tube. This one I've spiked with the dideoxy A, this one I've spiked with the dideoxy C. And so then when you look at what's in the tube at the end, for the A tube, you should get every single possible length of DNA, a copy of the DNA sequence. Where I get truncated, I get stopped at an A in the sequence. So in my sequence up here doesn't have a whole lot of T's in it, it only has 2 T's in it. And this sequence will be complementary to that. And so where I see a T right here, I should see some sequences get truncated into dideoxy A. And so that happens for this position, and that happens for this position right here, where I get terminated into dideoxy A. So for my C's, in my template, anywhere that I have a G, I should see a termination. So I get this length, and that length everywhere where the complement comes up to the G in my template. And then for my G's, I'm going to look for C's in my template, and that's where it's going to get truncated. So I get every single possible length that ends in a particular nucleotide. Then you have your 4 reactions. You just run this out on a gel. And you separate by size. And since I have every single possible length between my 4 tubes, you can read it on the gel starting at the bottom with my shortest fragment. And since we're sequencing 5' to 3', the bottom of the gel is my 5', and I just read up. So that's a C, C, A, T, C, G, T, T, G, A. And that is the sequence of my DNA complement to my template DNA, and that was what was, I think, throwing most people on their question on something, because they want the sequence of the template. So you have to take that C, C, A, T, C, G, T, T, G, A, and write out the reverse complement 5' to 3'. So A would be a T, G would be a C. So T, C, A, C, G, A, T, G, G. The answer for this one is the template. So it's the template right there. Okay. That's the cartoon gel. This is what they really look like. I'm happy to say I actually ran one of these in grad school. But then it was becoming common enough that you could outsource to other labs. And in Colorado, like the second year I was there, they established a DNA sequencing lab. I just sent my samples out and someone else did it for me. Because what a pain, my goodness, to get this to work just exactly right. This one would have been a case where your A's, probably in all of your reactions, were P32 labeled. And so this was radioactive, and you slap a piece of film on it and exposes the film, and you can see all the bands. But you just read it up, and this is a little harder to read, right? Really harder to read. So a C and an A and a G and a C, and then a T, a C, and a T, and a T, and an A. But you just read it off the gel. Yeah.
Do you write that in? You do write the primer in as well in reverse.
Well,
right?
yeah, I mean the primer is part of your sequence,
okay.
but frequently your primer is going to emphasize the region you care about. So.
Okay.
All right, that is with the gel, doing this kind of big gels. This is the type of sequencer we have here in the department. In the second semester, you'll get to sequence DNA using one of these. Not radioactive, but it will be a fluorescently labeled primer that we use. That is now outdated. DNA sequencing technology is just changing. And this is still, like I said, new to this edition of the book, but already 10 to 15 years behind time. Rather than doing 4 separate reactions and running a gel, which takes forever to set up and run, it's an all-day event. And fortunately for you, I will be setting up the gel for you. And you just have to run the PCR reactions for that. Rather than doing that, they started doing fluorescently labeled nucleotides where each one fluoresces at a different color. You excite it with a laser and it fluoresces a different color. And so rather than setting up 4 separate reactions, they just do one reaction with all the different colored dideoxys in the same tube. Same idea, you get this random incorporation, and with the entire population, you should get every single possible fragment length where it's terminated. Then you take the entire mix and you put it over a capillary electrophoresis column. So this is also a gel, but rather than a slab gel, it's a thin little column, and then you place current across it, and the things migrate just as usual. So your little stuff comes off first, and then you just stick the laser in a detector down here at the bottom, and it excites, and then it looks to see what flashes of color are detected. So then you can get your CBC, AAT, C, G, T, C, G, A for the different colors. It's a lot faster to do, and gets it to the point where the procedure is more automated as well, and these capillary columns can be reused. You don't have to pour a new one every single time you do a customizing run. So that is more typically what's done today, except that the next generation sequencing has blown this out of the water, and it's not in your textbooks because it's still too new. I'm sorry. That's typically what you'd see. So if someone was doing sequencing by this method with a little detector, you would just look to see for intensities, fluorescent intensities that come off the column over time, and so you just get our sequence data like that. I wanted to show you that. So with next generation sequencing, where they can sequence on a chip now or a flow cell, it has become much faster and much cheaper. So this was an ad I got actually by email last year, and I'm sure the price has come down. It was $1,000 for your exome, and that is going to be all of your kind of open reading, printing, exome type pieces. So not the introns of your DNA, but just the stuff that gets expressed in the cell. Or $6,500 for your entire genome. And, this is a little different for the personal medicine because this is a research ad, because they're expecting people to send stuff in from other organisms. But like I said, the one I got from the health every day was the personalized medicine. And that's in this range. If they're just doing exome again, which might be all you need, it's just going to be over a couple $1,000. It's not that much anymore. Should you want to signatures in So Human Genome Project, 13 years. Took 13 years to do, to sequence the entire human genome. So 10 years for initial draft, plus 3 years to finish the sequencing, and it cost $3 billion to do. But this project is what enabled this new technology to come out for the sequencing, which is why now you can do the entire human genome in like 3 days. It's done in 3 days. And it's only going to cost you a few $1,000. It's all, not $3 billion. So this is huge changes over the last 20 years. Okay, so next generation sequencing is a little more complicated, and you're going to read about that on Monday. Okay. So just a reminder that for gels, you read bottom to top, 5' to 3', to get a sequence that is complementary to the template. Okay, because that question is now back under the skills checks, so you don't have to answer it. So talk a little bit about primer design and what makes a good primer. So since we talked about PCR already and sequencing, you also have to have a primer for that. So this is something that needs to be complementary to the region of DNA you want to sequence. But you want high specificity, or we're going to talk about stringency. In the PCR reaction, you don't want to have your primer binding to any old sequence. It needs to be specific for one sequence in your sample. And it needs to have a TM, which is a melting point. Between your annealing and your extension temperatures. Or let's say, or at, let's say, ideally at extension. So anything between your annealing and your extension would work, but ideally it'd be right at your extension temperature. So a TM, this is the temperature at which 1/2 of your DNA is denatured. Okay. And so for DNA, we're talking about going from double-stranded DNA to single-stranded DNA, where you pulled apart all those hydrogen bonding interactions. If you look at the denaturation of DNA, and again, the OBS is just some observable, something I'm looking at. So maybe it's a UV or fluorescence or however you're doing detection. What you see is actually kind of cooperative looking. Altered denaturation. And your TM is the point where you're halfway between as your double-stranded DNA, and this is your single-stranded DNA. Your TM is that temperature halfway in between the two. Calculating a TM. There are better, more accurate ways to do this. We're just going to use the rule of thumb method. You get 2 degrees Celsius for every A to T base pair PP. And you get 4 degrees Celsius for every GC base pair. Okay? Why is that one higher?
3 bonds.
Okay, so that one's higher. So if I have my favorite primer, GAGTACA. Ha 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 ha. Right, okay. Nobody gets it. How many have seen that movie? Thank you.
What movie?
How many are going to go get this movie over the weekend and watch it?
What movie? What is it?
No, it's King Gattaca. This is terrible.
Oh.
Alright, that's some very relevant questions for today's society of sequencing DNA and what information should be private and information should be made public. And
So we should watch it in class? On Wednesday.
Okay, that's my favorite primer. So rather
That's our
than
test. And 2 questions.
I have two GCs, and I have 1, 2, 3, 4, 5 ATs. So I'm going to take 2 times 4 degrees C, and 5 times 2 degrees C is equal to 8 degrees C and 10 degrees C, and then I just add them together for an 18 degree C. So my TM is 18 degrees Celsius. That's pretty low. So maybe it's not my favorite primer after all. This is pretty low. So I'm going to primer. It doesn't mean that it won't work entirely.
What's a normal range?
Sorry?
What's a normal range?
Yeah, we're going to put that. Good primer should have greater than 90% of DNA, greater than 90% of the primer annealed. To DNA template. At the annealing temperature. Okay, what was the annealing temperature? Yeah, sure. So somewhere 50 to 55 degrees, somewhere in there is our annealing temperature. So you want 90% of your primer to be annealed at the annealing temperature. If my TM is 18 degrees, is 90% of it going to be annealed at a temperature of 50? No. So basically, I need to see my annealing temperature below the TM for my primer to guarantee that I have enough of it annealed at the annealing temperature. So you have to make sure your TM is at or above the annealing temp. And if it's at, then it's going to be 50%. Above is better. Okay. People get away with primers that are down in that 50-degree range, but it just doesn't work quite as well than if you can get more of it to anneal. So a good primer should be that, should anneal, mostly anneal at the annealing temperature, and then you want approximately 50% anneal at the extension temperature, which is what? Yeah, somewhere around 72 degrees Celsius. Okay. So if I want a TM near my extension temperature, then what do I want my TM to be? Around 72. Yeah. Usually a little under. This is what people pick, but usually it's better if you're closer to the extension temperature than you are to the annealing temperature. But you've got to be somewhere in between the two in order to get this amplification to even work. This provides stringency. We want to amplify one sequence. We don't want our primer to randomly bind to other regions because there's some base pairing happening. We want it to be very specific for our region of DNA we want to amplify. What will happen if the TM of the primer is too high? nothing will be amplified. As well as random things are going to be amplified. So you just have a big mess. You can't make heads or tails of what you're getting. Because, when you raise it to 95 degrees, it's possible to pull apart the entire double-stranded DNA of your big fragment. So you're going to be able to pull away your primer. So you're still going to be able to anneal and pull it apart and go through your random cycles. But if your TM is too high, what you start finding is that it can bind, it's big enough and long enough that it's going to start binding to other sequences, where maybe you've got 20 nucleotides that match exactly where you want it, but then there's another region in your DNA where 10 of those also kind of match, and then you get hydrogen bonding, maybe not standard Watson-Crick, but you get hydrogen bonding. And because there are 10 of them, the TM is high enough to be in that annealing temperature range. And so too high, you get random amplification. Okay, so what happens if your primer is too low? Your TM is too low. Then by the time you raise the temperature back up to your extension temperature, nothing's going to be annealed anymore. You still need around 50% anneal at the TM in order to see extension. So that would be okay. This chapter hints at a lot of different things you can do with DNA, if you know it. But we don't have time to talk about all of them. So there's the section about different DNA manipulation techniques. We've talked about how to sequence DNA. We've talked about how to amplify DNA because you have to understand this area to know how to sequence. Question?
If you had Gallica as your timer and you wanted to raise the TM, would you just add?
You just add more.
Okay.
There's one more technique I want to talk about because it's kind of an important one in terms of reading articles later this year. So site-directed mutagenesis. This is the technique made possible by PCR. This is where you change the sequence of a gene to produce a protein with an altered amino acid sequence. This is typically done with a gene that's been put into a plasmid. In bacteria, because it's just so much easier to work with. So you can do this with human genes and human proteins, but it's easier if you stick it into a bacterial plasmid and work with it in bacteria. Basically, you have two mutagenic primers. For both the sense and the antisense strand. When these are expanded by PCR, the entire plasmid is copied. And protein function and structure. Because we can just go into that sequence and we can change it around and see, well, okay, what happens to my protein? Does it still hold up? Is it still functional? Is it an enzyme? Does it still catalyze the reaction it's supposed to catalyze? So it's a way of doing protein studies where we can purposely make mutations. So, picture. So this is my bacterium. Inside is a circular piece of DNA, my plasmid, and it's showing the codon that codes for, what was it originally? Alanine or GTC. So it's originally an alanine. GTC codes for a valine. You have to amplify the plasmid by just growing out a bacteria culture. And so then you've got your millions or billions or whatever little bacteria cells. And you lyse them open and you purify the DNA. So now you have this little piece of DNA. It's being shown here as single-stranded because in your PCR reaction, you're going to amplify with a primer that's going to anneal to this. So here's my little mutagenic primer, and here it's only showing for one sequence, but normally you would do this with two primers, so you amplify both sequences. And so GCC codes for alanine instead. So the primer binds in the region around the codon I want to change, and it doesn't base pair exactly where the mutation is, but on either end of it, so this little part right here and here, that does base pair very well, and it has a TM within our range that we want to use, and usually somewhere between 60 and 70 degrees. And so even though these don't match, there's enough base pairing going on that your polymerase can extend it. And so then you just do the PCR reaction where you extend your primer all the way around, you copy the entire plasmid, and you do that over and over and over again, and eventually pretty much everything has the mutation in it, except for the little bit of template you studied with, and it actually tricks to get rid of the template at the end. And so then when you take this plasmid and you put it back into bacteria, any of the ones that picked up this primer with mutation in it is now going to make alanine instead of valine. So this is a really powerful tool to incorporate mutations that we want to look at in protein. PCR. I don't know if it is well about that anymore. Carey Mullis is the guy who came up with this, and he got the Nobel Prize in 1993. Anybody know anything about him?
Yeah, same.
You weren't born then?
We were born that
You
year.
were born then? Okay, so the story about Carey Mullis. He went to Berkeley, got his PhD at Berkeley. He worked in biotech down in California. He is a surfer, and he likes to ride motorcycles. And supposedly he came up with the idea of doing PCR while riding down, what is it, the Pacific Highway on the California coast, 101. And it just came to him, use a thermophile polymerase, and then you can amplify fast in the test tube. So, that's all brilliant, but the guy is kind of a whack job. He's a surfer from California. Well, he's a surfer from California who was heavily into using LSD. And so he got the Nobel Prize, but then people would invite him to come to conferences and give talks, and he started giving these weird talks. So he believes that everyone should use LSD. He thinks it opens your mind. He believes in astral projection in the astral plane. He believes in ESP. So he'll go to scientific talks and talk about things like this. He believes in aliens, and he says he's met one in the form of a fluorescent rectum.
I want to meet this man.
So, all of those things you can kind of this guy is just on his rocker and he's used too much LSD, but he's also said some things that have really infuriated people. So he believes that AIDS is not caused by HIV. But that's not true. Not possible.
Science.
So his kind of odd. He has an autobiography, Dancing Naked in the Minefield, if anyone is interested in reading it.
He probably would be the best professor. At your homework, try LSD. And then write about the fluorescent raccoon. That's biochem class.
Okay, so we have a couple things to watch.
It's your song. Your handwriting.
I thought we carried out a table saw. These are not serious at all. And I'll apologize ahead of time because one of them I can't pull a screen very good. Where's my sound?
You just turned it down.
Nope.
Oh, it has mute.
Oh, that was.
There was a time when
These are apps, just so you know. There's someone thermocyclers, there's someone.
there was a time when to amplify DNA. You had to grow tons and tons of tiny cells. Oh, little long can they get it done to carry more. Said you can amplify in vitro just as well.
Is that it?
Nucleotides and polymer bases too. Naturing and kneeling and extending.
Look at that spider vest. You're just jealous.
We'll have to do like the song for our vision to get extra credits.
That would be perfect. When you need to find out who the daddy is.
Scientists out there doing PCR, Biolab salutes you with the all-new 1000 series thermal cycling platform.
I want it.
Let's buy it.